and welcome everyone to this video podcast for evolution and diversity. In this video podcast, we're going to focus on the fungi. And in doing so, we're going to cover these 7 objectives. I want to begin by introducing you to the different groups of fungi, talking about some of the general characteristics that fungi have, the structure of fungi. We're going to talk a little bit about how fungi feed, their life cycles and reproduction, fungi as a phylogeny, how do they relate to animals or plants, and the different interactions fungi have with other organisms. So let's go ahead and get started with this list of different groups of fungi. All right, there are 6 different lineages of fungi, and let's start off with this first one, the Microspheridia. Now, all Microspheridia are parasites of various animals. Now, they will usually only infect fish and insects. However, there are some that will infect humans. They can make us sick, but they are particularly dangerous for those who are immunocompromised, so individuals who are battling cancer or maybe someone who has AIDS. This second group, the Catrids, a lot of them are parasites of plants and some animals like mosquitoes and amphibians. They also are one of the large culprits in the decline of amphibians across the world. Now, they don't all form parasitic relationships. Some of them have mutualistic relationships and live in cows and deer, and others have roles in decomposition, particularly that of plants. The third group are the Zygomakata. These are often what we associate with food molds, so I'm sure you've seen these before at some point. Some of these are used in producing fermented foods, as well as fermented beverages. The fourth group are the Zygosporangia. These will often form mutualistic relationships with plant roots. They are also important in the ecology of prairies and tropical forests. The last 2 groups are shown here, and let's talk about the Basidia first. These include the mushrooms and puffballs. So you've certainly seen some of these, and I'm sure many of you enjoy mushrooms, so many of you enjoy this group of fungi. However, some are also parasitic to plants. Zygomakata, these are the traditional yeast that you might use in baking or brewing, but they also include morels and truffles, so some other yummy tasting fungi. They also include a group called the lichens, and these are very sensitive to air pollution, and so because of that, they are often used as bioindicators to test the degree of pollution in a particular area. All right, now let's talk about characteristics of fungi, and I'm only going to mention a few here. The first is that fungi are heterotrophs. There are no fungi that are photosynthetic. In getting their nutrients, they often serve as decomposers, or they get their nutrients through symbiotic relationships. These symbiotic relationships can be a variety. They can include parasitic relationships where the host is harmed, or they can be mutualistic relationships where both the fungus and the host receive some benefit. The next characteristic I'm going to mention has to do with their mobility. And this characteristic is that fungi are sessile. And this term refers to organisms that are anchored to a substrate. And this substrate is often a source of nutrients. And the next feature I want to talk about are their cell walls. And their cell walls contain a structure called chitin. This provides a great deal of strength to the cell walls. Interestingly, this is also a component, chitin, that's found in the exoskeleton of insects. And the last point I want to make here is that fungi are not vascular. They don't have the vascular system that we just learned about in the gymnosperms and the angiosperms, and they also don't have the vascular system that we will learn about in the future when we talk about animals. The first thing I want to say about their structure is that yeast can exist in 2 different growth forms. So these 2 different forms include the single-celled form, much like you might see in the various yeast species, the ones that are used in making bread or making beer. The other form is the multicellular filamentous form. And I'm going to show you a picture of this in a moment, but I want to introduce a term for you, and that is the term of Mycelia. When you see a picture of this multicellular filamentous form, you'll see very large filamentous structures. And these are what we refer to as the mycelia. I'm going to show you in just a moment pictures of these, but I'm also going to show you a video just showing you how these mycelia can grow towards food. So it's very important for these yeasts to use these mycelia to find food. All right, let's look at the video. As you can see in this picture here on top, we see the single-celled form of fungi. And these are examples of yeast. These are, in fact, examples of the common baker's yeast. On the bottom is this very large filamentous structure that we call the multicellular filamentous form. All of these filaments that you see here together, this is called the mycelia. In a moment, we'll introduce some other terms associated with mycelia, but for right now, just know that this is an example. This is a picture showing mycelial growth. And it's also important to remember that the mycelium is multicellular. Let's look at a couple videos. All right, in this video, you're going to see the growth of the mycelium moving towards its food structure. And you'll see it moving pretty fast, but this is a sped up version of its growth. It almost looks like a spider web, but these are the mycelium growing. Here's another example of mycelial growth in the direction of food. The ability to move towards this food in this unified fashion helps the fungus absorb a lot of nutrients, and we'll talk about that in a moment, how it is able to digest and absorb these nutrients that it encounters. Okay, now I want to talk about what hyphae are. So hyphae are the long, narrow, frequently branching filaments that make up the mycelium. To look at a rather crude picture here of mycelia that's growing, as I said before, probably towards a food source, it contains several different units of hyphae. Okay, so let's pull out one of these simple structures here and expand it a little bit. Now, in most cases, these hyphae are separated into different cells with these different structures that we call septa, or septum for singular, septa for plural. Now, each septum will contain very small pores. These pores allow the mixing of cytoplasm between neighboring cells. So this allows for a nice way to move nutrients from one part of the mycelium to another part of the mycelium. I want to show you a quick picture just to show you how densely packed these hyphae can be. So this is just a picture of a mushroom, but you can see how tightly packed these hyphae are within the mushroom stem and the mushroom cap. So what you eat when you're eating a mushroom are these hyphae structures. And it's really important that these hyphae are tightly compacted, as we'll talk about in a moment, because it helps conserve water in it. Because once they're out of the ground like this, they have an increased chance of drying out. Now, some fungi will not have septa. Rather, they'll have a continuous cytoplasm where the nuclei in the different cells freely mix. I didn't draw nuclei in the last one, I just realized, but they would have been one per cell. We call fungi that have no septa coenocytic. So they are just this single, very large, multinucleated cell. And here's a nice picture of what these coenocytic hyphae might look like, where the green stained structures are the nuclei, and you can see that they're not separated into distinct cells by the septa. Okay, let's move on to our fourth objective for today, and that is how do fungi feed? So let's draw a hyphae here, And we'll draw the different septa in it. So remember earlier I said that these hyphae grow towards some kind of nutrient. And I'm just going to draw the nutrient as this big circular mess here. And when this hyphae extends towards this nutrient, a few things happen. The first is that it secretes digestive enzymes. So it'll secrete from the hyphae various digestive enzymes that will digest this nutrient. And so this is an example of extracellular digestion because it occurs outside of the cell. Two enzymes I would like you to know that are part of these digestive enzymes are Lignin Peroxidase. And when we talked about the vascular plants, we talked about the importance of lignin. So fungi that secrete lignin peroxidase, they are going to be digesting those lignified structures in plants. So these are very important in the fungus's role as a decomposer. The second one I want you to know are enzymes called cellulases. So plants have a lot of cellulose in them, and fungi, when they feed on these plants, will release this enzyme called cellulase, which will digest the complex carbohydrate of cellulose. After it has digested the nutrients on the outside of the cell, the next step is absorption. Nutrients digested, and then once digested, they come into the cell. And now once they're in the cell, because of these pores, these nutrients can be passed down between the different cells, which will aid in the continued growth of the hyphae. Now I'm going to come back to this ability of the hyphae to absorb nutrients in just a moment. However, I do want to also mention another really interesting thing that some fungal species use their hyphae for. And as you can see in this picture, the hyphae can be used to capture prey and then feed upon that prey by absorbing nutrients from it. So in this picture, what we see is this nematode here. This is just a very small worm along the red arrow here. And now the hyphae I'll show in blue here, but it extends all the way up here, and you can see how it wraps around the nematode and forms this structure, as you can see here, that kind of pinches around the nematode and begins to digest nutrients from that nematode and absorb it into the fungal species, ultimately killing the nematode. Okay, now let's go back and talk about why fungi are really good at absorbing nutrients in their environment. So what is it about a typical fungus that allows it to bring in so many nutrients? And one of its most successful features is that the fungus has a very large surface area. Because of all of these branching structures found within these hyphae, that increases the overall surface area and allows it to move in more directions, but once it finds the nutrients and begins to digest it, it really aids in the absorption of all the nutrients. So this surface area is incredibly important. But there is a drawback, and I alluded to this drawback a little bit ago when I showed you the picture of the mushroom. And the drawback is that this increased surface area increases the chance of drying up. Now, what happens when these hyphae begin to dry up? Well, one thing that they will do is they will produce something we call a spore. And these spores are released. And I'm going to wait for just a little bit to describe what spores are and what they do, as that leads us really nicely into our next topic of life cycles and reproductive structures found in various fungi. All right, let's go ahead and move on to our fifth objective for today, and that is to describe the fungal life cycles and reproduction. I want to begin by talking about what spores are. And I briefly introduced these to you on the last whiteboard, but it turns out that fungal reproduction is really all about the spores. The spores are really important in the entire life cycle of fungal species. Spores are reproductive cells. They're produced through asexual and sexual reproduction. they are resistant to drying out. And I said it when I introduced spores here, but let's put it in writing. Spores are the fundamental reproductive cell in fungi. Okay, let's continue this discussion of spores. Let's begin by drawing a part of a mycelium here with these few hyphae on it. Now remember, this mycelium is made up of many cells separated by the septae. I just didn't draw it out here in this particular picture. But each of these cells, I'll just draw one like this, is a haploid. There's only one copy of the chromosomes in it. As the mycelium continues to grow, it's growing by the process of mitosis. Taking a 1N cell that's in the hyphae here and generating a new 1N cell, allowing it to get longer and longer with each division. Now at some point, at the ends of these hyphae, some spore-producing structure is formed. And from this spore-producing structure, spores are generated. And these spores are then released. And they're carried to a new location, typically just by the wind. In fact, it is very common for these fungi to produce many spores. And in fact, a typical air sample might actually have more spores in it from fungi than pollen from flowers. So there's a lot of these spores in the air. Now these spores are going to land somewhere eventually, right? And they're going to begin to germinate, particularly if they land on food or near food, if they can sense food. And they're going to begin to grow towards the food. Now at some point, the food's going to run out. And then once again, it'll begin to generate these spores and release the spores. I'm just drawing it in a different color so we can keep the different fungal mycelium separated here. So it'll release these spores, and once again, these spores will land somewhere and then begin to germinate and grow these hyphae as long as they land near food. Now, if it doesn't land near food, remember that these spores can last for quite a long time. And depending on the species, it varies on how long they can last. But certainly in some species, they can last several years. And I should also point out here, I said it here, but remember, as these spores germinate and begin to form these hyphae, that's all growth by mitosis, going from 1N to 1N. And same thing here. These are growing by mitosis, 1N to 1N, haploid to haploid. Now, what I've described here is asexual reproduction. All fungal species will have this form of reproduction, where they don't require a different fungi to mate with. I should point out that while every fungal species can do this asexual reproduction, there are two species that, to our knowledge, only use asexual reproduction. That is, we've never seen them participate in sexual reproduction. And these two species are Microspheridia and Zygomakata. And lastly, I want to introduce a term for you, and it is called conidia. And this simply is a spore produced through asexual reproduction. Okay, so let's talk about sexual reproduction in fungi. And I want to begin by talking about the Catrids. And I want to talk about these first because they are the exception to what we'll see with the other fungal species. And they are the exception because they produce sexual gametes. However, I should point out that these sexual gametes are not egg or sperm. It is true that the sexual gametes do have a female gamete and a male gamete, but they're not egg or sperm. A couple things set them apart from an egg and a sperm, and that is that both the female and male gametes are motile. Another difference is that the female gamete is only slightly larger than the male gamete. And I don't have room to mention it here, but Catrids are also the only fungal species that has a alternation of generation. So let me just write that up here. I'd like to show you a figure showing Catrids' sexual reproduction. So in this figure you can see the alternation of generation going from the gametophyte Mycelium to the sporophytic Mycelia. We see a similar trend that we've seen before where we have one N for the gametic Mycelium and a 2N for the sporophytic Mycelium. The gametes are produced from the gametophytic Mycelium. The male and the female gametes will fuse, and again, remember I said they're both motile, so that's why you see the flagellum on it. And I said that the female gametophyte, and I said that the female gamete is a little bit bigger than the male gamete. When we're looking at egg and sperm, remember that the egg is very much larger than the sperm. At any rate, these two fuse, and then they will form this zygote that we've talked about before. So this is the fertilized egg, a one-celled embryo. It will then divide by mitosis to produce this sporophytic mycelium, which will then generate spores, and these spores will be released, just like we talked about, and then they will continue to divide by mitosis, forming this gametophytic mycelium. And then the whole process starts again. You'll see in just a moment that other fungal species accomplish sexual reproduction using a slightly different mechanism. So the last three fungal lineages that I want to talk about are the Zygomakata, Basidomycata, and the Asexual. All three of these will undergo sexual reproduction, but they will not use gametes. What I'm going to do is go through steps describing sexual reproduction in these different fungal species, and then I'm going to go to a larger figure to walk you through it. So this first step is when two hyphae from different individuals grow together and the cells fuse. So you might have one hyphae growing in this direction and another hyphae growing in this direction. So as these hyphae come together, at some point those cells are going to get close enough to each other where they can fuse. And so I'm just going to draw this up here, what these cells might look like, and I'm going to draw them much bigger than what these hyphae might suggest they would be. But these cells I'm drawing here would be found right at the tips of them. And as they come together, they will fuse with each other. And you would end up with a cell that looks something like this. So this fusion of the cytoplasm of different individuals is called plasmogony. Then our next step is going to be when the nuclei fuse. So when this cell here undergoes nuclear fusion, the two nuclei come together forming a single 2N diploid nucleus. And so that's nuclear fusion. When this happens, this is called karyogamy. The fusion of two nuclei is called karyogamy. So when the cells fuse and the cytoplasm mixes, that's called plasmogony. And then when the nuclei fuse, that's called karyogamy. I'm going to throw one more term at you, and that is the term heterokaryotic. And I've kind of ran out of room here, so I'm just going to write it down here. But a heterokaryotic cell is what we would call this cell here. It is the cell that results after plasmogamy. It is a cell with 1N plus 1N, two separate haploid nuclei. We call that a heterokaryotic cell. Now on the last whiteboard, when I drew this, I showed that we ended up with a diploid cell that came together because of these two haploid cells merging into each other. I didn't say it on the last whiteboard, but this is a zygote. It's formed not by two gametes coming together, but because of two haploid cells that run into each other. Two haploid cells that are not gametes. So this zygote, which is 2N, will undergo meiosis to produce 1N, that is haploid, spores. And it will release those spores, as I'll show here in just a moment, like so. But it went through meiosis, so they're no longer diploid, as I say over here, but they're haploid spores. And they will move on, and just like we talked about before, they will land somewhere, and when they land, they will begin to grow by mitosis into a haploid mycelium. Throughout sexual reproduction in fungi, only the zygote is diploid. They're haploid before they fuse, and then when they release those spores are haploid, and they will grow and form into these mycelium as haploid. Okay, now that I've gone through these different steps showing sexual reproduction, I want to put it all together and go to the figure that's in your book. And so here what we see are two hyphae coming together, and they show them as being different because you see a darker nucleus in this one and a lighter nucleus in this one, just to kind of distinguish the fact that they are different individuals. And remember I said when these cytoplasms fuse, like we see right here, that is called plasmogony. This cell, as it exists right here, I'm going to circle it, and every time it divides to produce another cell like that, they are called Heterokaryotic Mycelium, because the nuclei have not yet fused with each other. So they are 1N plus 1N cells. Now at some point, these nuclei will fuse, as is shown in this cell here, and they show it with a gray nucleus, indicating a darker nucleus and a lighter nucleus came together, and now you get gray. So when they come together, they form this diploid zygote. And the fusion of these nuclei we call karyogamy. This diploid zygote now will undergo meiosis, producing haploid spores. You see them right in this spore-producing structure, 4 of them. Every diploid nucleus is going to produce 4 haploid spores. And then they will be released, they will blow in the wind, they will land somewhere, and wherever it is that they land, they will begin to form a new mycelium, a haploid 1N mycelium. And then the whole process repeats itself. Now, one more thing I'm going to talk about as we think about sexual reproduction are mating types. Remember, fungi don't have male and females. So how is it that when we had this one hyphae growing towards a food source or towards something, and this other one moving also in that same direction, how did they know that they were different? How did they know that they could fuse with each other? 2 green hyphae would not fuse together, and 2 red hyphae would not fuse together. They fuse together because they are different mating types. And these different mating types have different names depending on the species of fungi you're looking at. Sometimes we call them plus minus, we might call them A and alpha. There's a lot of different ways to talk about them, but they are recognized as being different. These different cells of these different mating types have different sets of alleles, so this helps with genetic diversity by making sure you only allow mating to occur between different mating types. So an increase in genetic diversity. Now, an interesting thing about fungi is that these mating types sometimes exist as 2 different mating types. But more often than not, they may exist in many different kinds of mating types within the same species. One particular fungi is thought to have about 28,000 different mating types. Now that's an extreme. They don't all have that many, but that's just to give you an idea that the number and variety of mating types in some cases can be as different as the number and different types of alleles that are present in the different individuals within a species of fungus. Now I want to talk about reproductive structures in fungi. We've already talked about how the 2 hyphae meet and they fuse their cells to mix their cytoplasms, and then how the nuclei fuse, and then you end up with this zygote, and then the zygote produces the spores. And so when we talk about reproductive structures in fungi, what we're really going to be focusing in on is the spore-producing zygote and how it is formed. And to some extent, we'll address the spores that are produced as well. There are going to be 4 of these that I'm going to talk about, and your book goes into great detail with all 4 of them, but I'm going to break it down and let you know exactly what you need to know. So first, let's list the 4 different reproductive structures. The first are swimming gametes and spores. The second is Zygosporangia. As the name suggests, we typically see species with Zygosporangia in the lineage of zygomycetes. The third type of reproductive structure are Basidia. And as you might predict, species in the lineage of basidiomycetes will typically have these structures. And our last one are the Asexual. And as you might have predicted, the ascomycetes are the ones that traditionally will use Asexual. So I'm not going to say a whole lot about the swimming gametes and spores, except to say that we see these in the Catrids, as we've already talked about, and we will see them in other species that are aquatic, where having a swimming gamete or a swimming spore makes sense. Let's talk about the second one now, the Zygosporangia. Zygosporangia will form when we have different individuals from the zygomycetes come together. They form these structures at the end of their hyphae, and that come together and fuse and form the structure in between. And the structure in between is the Zygosporangia. The Zygosporangia then will generate spores that can be released and ultimately germinate and form new hyphae. I'm going to go to the figure and show you how this fits into the life cycle. So you see here, this image here looks very similar to what we saw with the generalized sexual reproduction. So you don't need to memorize this figure. I'm just showing it so you have a reference for it. You have the two hyphae from different individuals coming together here, and then they fuse during the process of plasmogony. They form this heterokaryotype that we talked about before. That is the stage before it becomes a zygote. So at some point the nuclei fuse, and when they fuse, they produce the zygote, and then the zygote here, which is 2N, will undergo meiosis and produce spores. And then these spores will land on some nutrient source, like this piece of bread here, and begin to form hyphae. Now these hyphae, they can undergo asexual reproduction, where they will just release spores from the hyphae, or if two hyphae come together, like we see here in this box, they will once again form this Zygosporangia, and the process continues. The main point of this figure is to show you where the Zygosporangia forms and how the Zygosporangia will ultimately release those spores to continue the life cycle. All right, let's talk about the Basidia. Remember, the Basidia are produced in the basidiomycetes, and the basidiomycetes represent the mushrooms as well as other fungal species. So I'm going to attempt to draw a mushroom here. It's probably about as good as my human stick figures. So here's my mushroom. And remember, hyphae will form up the stem here, and then they are packed very tightly within the mushroom cap. These hyphae that make up the mushroom cap will fuse with each other. And we're just going to draw them like this. And when they fuse, they form these club-like structures shown here. And together, these are the Basidia. Each individual structure here would be called a basidium, because that's the singular form of Basidia. Nuclei then will fuse in these Basidia to form a 2N zygote. These Basidia then, each one of them will release spores, and these spores will ultimately land on some substrate, and the hyphae will grow from here, and then at some point you will form a new mushroom. Let me move to a figure to show you how this fits into the whole life cycle. Again, you don't need to memorize this whole figure here. I'm just mainly wanting to highlight where we see the Basidia. So here are the Basidia, and you can see how they look like these clubs. And remember that they originated from this cluster of hyphae that were found in the cap of the mushroom. Within these Basidia, the nuclei will fuse and it becomes the zygote. The zygote will then undergo meiosis, producing spores, and then these spores are released. And when they land, they will generate new hyphae, and these hyphae will continue to grow and often will continue to grow as they merge with other hyphae through sexual reproduction until they can form this much more elaborate network of hyphae that then forms these mushrooms. All right, let's talk about our fourth structure, and that is the asci, or ascus as singular. Essentially what these are sac-like structures that form off of the hyphae. Similar to the mushrooms, the asci will form various kinds of structures, and there's a great variety of them, but one of these are these cup fungi, and essentially they look something like this, which in some ways looks like a mushroom, but it's just inverted. So like the basidiomycetes, the hyphae that are within this cap here are tightly compacted, allowing for the different individuals to undergo sexual reproduction. And within each individual hyphae, which I'll just draw like this, there will be multiple nuclei from the two different individuals. But remember, they had to be of opposite mating types in order for them to fuse. So essentially what we're looking at here is one large cell with a lot of different nuclei in it. And remember, these nuclei are all haploid, 1N nuclei. Now, two of them that have a different mating type will fuse during karyogamy. And when they fuse, they will become a diploid cell. And it will form its own cell here. So this is the zygote. The other nuclei are still here, they just haven't fused yet. When they do, they will also form a zygote. But each of these zygotes now will produce 4 haploid spores through the process of meiosis. Each of these spores then will land on a substrate, hopefully some kind of nutrient, and will then again germinate and begin to form hyphae until they ultimately form a new aggregated hyphae structure, such as we see in this. So again, now let's look at this figure here that shows you where the asci fit into the life cycle in a figure that's similar to what we've seen before. And again, remember that you don't need to memorize this figure, but it does help understand where they fit in here. So within the tightly compacted hyphae here, various hyphae from different individuals will fuse with each other. And when those hyphae fuse, they form this large cell here with the different nuclei in them. 2 of them from different mating types then will be enclosed inside of a single cell. And we call this the ascus here. And then the nuclei will fuse, forming the zygote, the diploid zygote, and they will undergo meiosis, producing the 4 spores. Now, in some species of Zygomakata, after the 4 spores are formed, they will go through an additional round of mitosis. And that allows them to create a total of 8 spores that are going to be released. Now, some ascomycetes don't undergo this extra round of mitosis and then just proceed to producing the 4 spores. But at any rate, regardless if it's 4 spores or 8 spores, those spores will land, they will begin to germinate and form new hyphae that will then continue to grow until they form this large cup-like structure. All right, now I'd like to spend just a little bit of time talking about the phylogeny of fungi. And there are 2 questions I want to address. The first is, are fungi more closely related to animals or plants? And this one we're going to do in class as it relates to a specific activity I plan to do. And the second question is, which fungal groups are monophyletic? So to address this question, let's look at a phylogenetic tree showing just the fungi. And we can see our 6 different groups here. We have the Microspheridia, the Catrids and the Zygomycetes, the Glomeromycata, the Basidiomycata, and the Zygomakata. And so the question is, which of these are monophyletic? So go ahead and hit pause and think about which one of these A through F are monophyletic. And then when you think you have an answer, go ahead and hit play again. All right, so remember that a monophyletic group includes a common ancestor plus all of the descendants from it, and only the descendants of that group. So for instance, the Glomeromycata, shown here, these are monophyletic because they have a common ancestor here, and then they have different lineages from that common ancestor. But only members of Glomeromycata are in this group here. It doesn't include anything else. So that's why it's monophyletic. Same is true with the Basidomycata. They have a common ancestor here, and it only includes members of Basidomycata. Doesn't include members of any other group. And lastly, down here, Asegomycata are also monophyletic for the same reason, because of this common ancestor only giving rise to members of Ascomycetes. So these three here are monophyletic. The Microspheridia, the Catrids, and the Zygomakata are all paraphyletic because they have a common ancestor here, but it includes members of all 6 different groups. So they are paraphyletic. All right, that ends this video podcast. There's a lot of information in this about the fungi. If you have any questions at all, please make sure you come talk to me or send me an email. If not, I will see you in class. Bye for now.